on personal genomes for people. And just to remind people that all these, this is pretty close to what we had last year. So you can just download the stuff from last year, and I'll repost the stuff from this year very shortly, and so forth, as well as the video and so forth. So I just wanted to say that we introduced the field of biomedical data science and bioinformatics to you in the first class, and you've heard a lot about the data in subsequent classes. I just wanted to give you a sense of the big applications for this discipline. And one of the applications, of course, is characterizing personal genomes, as we're going to talk about. But just let's talk about some of the applications. One application is really helping to design drugs. That's an important thing that people do. Now, usually this work is very structural. So people are thinking about structural modeling, docking, also screening a lot of compounds in silico and so forth. And so here we have little pictures of docking and so forth, which is something we're going to talk a little bit about in the class, but not that much. Another application, which we're going to talk a lot about, is sequence comparison and finding homologues. And let me say why this is very important. One of the most amazing things, as you know, about biology is we, being humans, share a lot of basic biochemistry and our genetic makeup with many other organisms. And one of the key things, of course, is we can't do experiments on people or rapidly screen things and so forth, but we can do this for a lot of other organisms. And so finding the homologs, particularly distant homologs sometimes, for all the genes in the human genome in organisms that are much more experimentally tractable, whether they're a mouse or even a worm or a yeast, is really, really useful for biology. And so there's a lot of interest in bioinformatics in trying to do these sequence comparisons very fast and very sensitively. A second application. So the third application I just want to point out is now that we're doing a lot of whole genome analysis, people are very interested often in characterizing the exact mutations in a person who is sick, and I'll tell you about that in a second, but specifically, if you have cancer, you have what's called somatic mutations, which are mutations that are not part of your germline. We'll talk a little bit about this. And one of the really amazing things is people have often been able to customize the treatment for a cancer patient based on the specific mutations and lesions that they have. And here's a very famous example of a drug, this Gleevec drug, which is targeting as very specific gene fusion between this BCR-ABL proteins and so forth. And people can really target this. This is for a particular condition. I think it's called CML, and they have a very targeted treatment based on the mutations. Okay. So another application I wanted to point out, which is related to this too, is we can look at someone's genome and we can find the genes associated with the disease. And a lot of times those genes give us very good hints as to what we should target for a drug. And a very good example of this is neuropsychiatric diseases. So these diseases, which are things like schizophrenia, autism, bipolar, are amongst the most heritable conditions, most heritable genetic conditions. Sometimes the heritability here is estimated as up to 80%. And now the interesting thing, though, is we don't really have a very good idea of exactly the mechanism. And this is really a contrast to cancer, where we understand really the pathways that are involved, or even heart disease, where we understand a little bit what's happening. For a lot of these diseases, we don't have a clue about the underlying basic biochemistry. But what we can do is, since they're so heritable, we can track through the mutations that give rise to those conditions and know, oh, those mutations are in particular genes, and then try to look at those particular genes as potential drug targets. So a lot of times, here's the bioinformatics is giving us hints as to where we should look to start designing a drug where we have no biochemical clues. And then the last application I'm just going to mention is what I call holistic personal genome characterization. I put this last, of course, is because it leads into what we're going to be talking about with Carl. And this is the idea of really looking at a healthy person and kind of building their whole data cloud, all of the information about them, their genome, and as we'll talk about, their phenome, other information, and looking at how it potentially changes over time, longitudinally. And, now it's not clear necessarily what all the actionable medical insights for this, but it's tremendously illuminating. And I think more and more people are going to want to know about their underlying biology and the data associated with that. And so that's another big application. Now I'm just going to introduce Carl here a little bit. So Carl is really one of the pioneers in this, I guess, world of exploring your genome. And he's really quite a pioneer. And he's sequenced his genome, as he'll tell you. And he's even kind of created I don't know how to put it, a whole series of stories about it. He initially called it the game of genomes, and you can read about it. He's done a spectacular job writing about this. And what we're actually going to do in the class is do some analysis on Carl's genome. And it's actually quite amazing to, I guess, meet the person you're analyzing. And for me, that's a huge thing. And I think for you guys, it's actually quite amazing. We're going to be talking about the trying to characterize the mutations in Carl and telling him what we can find out from them. And, he's interested in that because that's him. And we're interested because it's a person we know and so forth. Now, I just want to point out that we're not going to do this in this class specifically, but in past classes, another person we've looked at in detail is this guy here, Mike Snyder, who used to be a professor at Yale. Now he's a professor at Stanford. He's their genetics chair. And he's kind of went a little bit beyond even the genome. He has this thing he calls IPOP, which is, I think it is a harbinger of the future. It's basically where you create this data cloud about you. So he walks around all day with all these crazy sensors on himself, and he measures things and all. And you can imagine, in addition to having your genome sequenced, doing your transcriptome at regular intervals, doing your methylome, doing other types of omic assays, proteomics, metabolomics, and so forth, coupling this with, the very deep description of your phenotype from devices in your medical record. And this is something that we might be seeing in the future. Now, with that,